So you're going to find out that the story is a little bit more complicated than you can tell from just that title. So first I'll introduce you to the species that I'm talking about. There's Designatus quadrimaculatus and Designatus marmoratus. There are two stream salamanders that live in southern Appalachia in the family Phylogenomics. And as you can see, their ranges pretty much overlap completely, except for a small area in the north. I also wanted to point out that there's another species within this clade called Fulcurzi. It's a dwarf species, and I didn't study them for this project, but they will be included later, and I'll mention that at the end of the talk, so I just wanted to point them out. That completely startled, but that should have been pictures of the different morphologies that you can tell from those photographs at the bottom there. So marmoratus and quadrimaculatus have been described based pretty much on their morphology. So I'm just going to call them marms and quads from now on because they have very long names. So the marm on the left there has, their common name is the shovel-nosed salamander because they've got a wedge-shaped snout. And they also have a lot smaller eyes than the quads do. And there's also changes in their internal and external layer shapes. And all these together have been suggested to be adaptations to a more aquatic lifestyle. But I do find these guys under the same rocks in the same streams. So I'm not quite sure that that is an accurate portrayal of their lifestyle, but we'll find out more later. So the big picture with these guys that I was interested in was it looked like this was a case of sympatric speciation. Like I said, they're in the same streams, they're in the same region, and they seem to share the same niche, basically. So I was interested in what are the genetic mechanisms underlying that divergence. And as you'll find out, I didn't figure that out at all because it turns out when I was looking closer, they had a little bit of a complicated picture in terms of their phylogeny. So previous phylogenies were done with mostly mitochondrial DNA, and the results came out like this. So we've got marms in blue and quads in orange, and as you can see, neither of them are monophyletic. And I don't know about you, but I like my species to be monophyletic. So this is a little bit concerning to me about, well, this doesn't seem to appear to be two species. But we do have the case in Designatus that there is mitochondrial introgression has been shown to happen. So you have the entire mitochondria is switched from one species to another. So mitochondria is not the best way necessarily to look at speciation in this group. So we needed a genomic approach. There's also problems with the genomic approach though. They have large genomes, so about 15 gigabase pairs, and which is actually rather small for a salamander, but it's still fairly large. And there's of course no reference genome. I don't think there's a reference genome for anything in this family. And there's high diversity between individuals. There are other people in the same lab as me who are working with the same genus, and they've already done some work on the genetic diversity and showed that this is a lot higher diversity than you would see between individuals in other taxa. So we decided to use dd-rad. If you guys aren't familiar with that, you take two restriction enzymes, and basically you're chopping up the DNA. This is a very simplified version of it. And you'll size select and be able to get, hopefully, a number of sequences that align between individuals. But, like I said, there's high diversity in these guys. So, one of the problems we seem to have found is that it's pretty common to have mutations at these cut sites, in which case you won't have that same locus. So what's been happening is, and just to reassure you, like, we do have a good data set, but if you look between species within the same genus, there's basically less than 1% overlap in this guy. So that makes it incredibly difficult to include an outgroup because basically I'd be left with two loci. You're not going to build a very good phylogeny based on that. So we do have some things that we're going to work on that I'll talk about at the end to make that, to fix that problem. But for now, when you see a tree on here, it's going to be unrooted because we couldn't include those outgroups. So I did several analyses, So what I've done is I'm showing you the admixture results here on the map. So those are the sampling sites that I did. And each one might represent more than one individual. But the results across each of these analyses were the same. So what it turned out to be was that there were 3 populations that were identified, and there was very little admixture within the individuals. So there's only a few individuals that show admixture between these populations. And as you can see, the squares are quads and the circles are marms, and they are assigning based on geography, not based on species, which is not what you would expect if they were separate species. So we built an SVD cortex lineage tree to take a closer look at this. I just want to point out that on the terminal edges, like, we're not quite sure about the support there because there's quite a bit of differences between each branch. But the main split in the center there is 100% bootstrap support for those 3 clades. So basically, this is the same results that we got with admixture and structure, where it's showing 3 populations and quads and marks are interspersed. But there could be some grouping within that. So it could be that perhaps there was a split and there's quads and marms, and then they would cluster on this if we had some better results for the tree in terms of bootstrap support. So what that leads me to is our 2 extreme hypotheses. So it's a gradient, basically. So the most simple one is that these guys are representing 1 single species that has 3 populations, and then there's 2 morphotypes. On the other extreme end, you've got 6 populations, or 6 species. Each population that we've identified represents a species, and each morphotype represents a species within each population. So obviously there's things that could be in the middle, like for instance, we could just have 3 species, we could have 5 species, we're not quite sure. So what we did to test this is some species delimitation and tree building using Bayesian phylogenetics and phylogeography, or BPP. So we started out with the most complex hypothesis with you have to give it a starting tree, so it's a 6-step starting tree based on the SVD cortex species tree that we built, which I haven't shown. So I did quite a few things in replicate. I did multiple data sets. I varied the priors. I basically tried to break it so that I could have make sure the results are quite robust. And this is the result, which was kind of unexpected. So we have the first split here, and you've got the population Pisgah East. Everything, no matter what the morphotype, seems to be 1 species. Pisgah West, the same deal. But in Nantahala, there's quads and marms split. So this seems a little bit odd. I don't know about you guys, but I was a little bit confused about this. About how could you have these morphotypes representing species in one area, but in the other area they don't? So here's one possible hypothesis for this. So let's say they started off with a single population. At some point, that pan-mictic population splits. Maybe it happened in one event, maybe there was multiple events. But these guys become reproductively isolated over time. And then there's similar ecological pressures that cause them to develop into these separate morphotypes, but they're still a single species that are just showing 2 phenotypes. Over time in Nantahala, this becomes a fixed trait, so it's canalized, and now you've got 2 species down here, but that process of fixation has not occurred yet in these other populations. Oops. So that's one of the working hypotheses. There's quite a few more. So a few of the things that we're going to do so that we can work on this and get more robust results are, first of all, we need to fix the problem with no outgroups so we can root the trees, because otherwise we're not going to be able to look at the different lineages and how they diverge. So we're going to try to use a technique called rat capture. And so basically what that does is instead of a double digest, we're going to use a single digest. And then we're going to use probes to grab those loci, which hopefully exist, but we haven't been capturing the DNA salamander is just one of the morphotypes. So it'd be interesting to see how these guys might differ from the areas where they overlap in range. And we'd also like to include Volkert site in this analysis because it is a dwarf form. There isn't actually any guarantee that that's also a separate species. Right now it's listed as one, but they use the same techniques to list that as a separate species as we're seeing with these guys, and it turns out that they're mostly morphotypes. And the next step, once we get that better data set, is to do hypothesis testing. So we want to know not only are these guys actually separate species in one area versus another, but when did that happen? How did that happen? So basically the underlying mechanisms, which we will get to eventually, but this is initial work. So that's just a year's worth of work. We still have time to do some more. And I'd like to acknowledge all the people who helped me. It was my first year as a PhD student, so I couldn't have done as much as I did without all the members of the lab and the funding sources, and of course all the salamanders who very